Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Today we are going to continue with the chapter we started yesterday, and this chapter is concerned with the concept of formation of concentrated urine. And yesterday we discussed how the extracellular fluid osmolarity affects the concentration of the urine and osmolarity of the urine, and how dilute urine is formed and how concentrated urine is formed. And I mentioned that two requirements for the concentrated urine are the presence of the hormone known as ADH and the presence of a hyperosmolar or a hypertonic renal medullary interstitium. So the second requirement is hyperosmolar renal medullary interstitium. And to form a hyperosmolar renal medullary interstitium, there are two mechanisms involved. First, there is the countercurrent multiplier, and then there is the urea recycling. And to maintain the osmolarity which is produced by these two mechanisms, there is another mechanism in place which is known as the countercurrent exchange, which maintains the hyperosmolarity produced by the above mentioned two processes. So to give a brief revision of the countercurrent multiplier, you have to remember that when the fluid enters the nephron, it has the osmolarity of 300 millimole equal to that of the plasma, and the interstitium is also having the same osmolarity. But as the fluid moves along the nephron, and the part which is present in the thick ascending limb is treated on by the sodium potassium 2 chloride transporter, and it pumps out solutes from the nephron into the renal medullary interstitium till there is a difference of 200 milliosmoles between the nephron and between the renal medullary interstitium. Next, the descending loop of Henle is going to lose water, and it is going to gain the osmolarity equal to that of the renal medullary interstitium which was produced by the thick ascending limb. And then next column of filtrate is going to reach the loop of Henle from the Bowman's capsule, and it is going to have the osmolarity equal to that of plasma, which is 300 milliosmole. This fresh column of filtrate is going to push the fluid from the descending loop towards the ascending loop, and now the fluid which reaches the ascending loop is again going to be treated by the sodium potassium 2 chloride transporter, and the thick ascending limb is again going to pump solutes from the nephron into the medullary interstitium till there is a difference of 200 milliosmole between the nephron and the renal medulla. Again, the descending loop is going to lose water, and it is going to make its osmolarity equal to that of the renal medullary interstitium. The step is going to be repeated 4 to 6 times till the osmolarity of the deepest part of the renal medulla becomes as high as 1200 milliosmole to 1400 milliosmole. And you can see, as you move from the cortex down towards the medulla, the renal interstitium becomes hyperosmolar. It increases from 300 milliosmoles to around 1200 milliosmoles. This is known as the countercurrent multiplier mechanism. So I hope you understood this mechanism, and I hope this revision was helpful. If you still have any doubts, please go back to the previous lecture, and you can revise this whole mechanism in detail. And if you still have any questions, please leave them in the comment box. So now let us continue with the next part of this chapter. Let us now study the role of the distal tubules and the collecting tubules and ducts in formation of concentrated urine. So as you already know that the urine which is reaching the last parts of the nephrons is highly dilute. Why is it dilute? because we moved most of the solutes from the nephron into the renal medulla to make it hyperosmolar. So by the time the filtrate reaches the distal convoluted tubule, its osmolarity is very low. And as you already know that the thick ascending limb and the thin ascending limb and the early part of the distal convoluted tubule are absolutely impermeable to water. No water can leave these parts of the nephron. And to make it more serious, there is reabsorption of sodium and other ions from the early distal convoluted tubule, which makes the filtrate even more dilute and even more hypoosmolar. So this is the reason that the early distal convoluted tubule is also known as diluting segment of the nephron. So now the fluid which is reaching the last parts of the nephron is hypoosmolar. It contains very little solutes and very large quantities of fluid. We cannot allow these large quantities of fluid to be excreted in the form of urine because we want to form concentrated urine. Otherwise, the whole exercise we did previously for making the hyperosmolar interstitium is going to be lost, because we need to reabsorb all of this water which is present in the nephron. In fact, most of this water is going to be reabsorbed if there is present ADH, so that concentrated urine can be formed. So in the presence of ADH, your distal convoluted tubule, the last part of your distal convoluted tubule, your collecting tubules and your collecting ducts are going to become highly permeable to water and reabsorb large quantities of water, and the urine which is going to be produced is going to have very small volume, but it is going to be hyperosmolar as compared to the fluid. So it is going to have large quantities of solutes as compared to water, and it is going to be hyperosmolar, and it is going to have an osmolarity of around 1200 milliosmoles, which is equal to the osmolarity we created because of the counter current multiplier. I hope you understood. So let me repeat that your distal tubules and your collecting ducts and collecting tubules are going to help in the formation of concentrated urine by reabsorbing most of the water with the help of ADH. So finally, the water which is the urine which is going to be produced is going to have very little amount of water, but large quantities of solutes in it. So this is how concentrated urine is formed. Here you can see how in the presence of ADH, water is being reabsorbed from the late distal convoluted tubule, from the collecting tubules and the collecting ducts, and as you go down the length of the nephron, the urine becomes concentrated because of the reabsorption of water until the osmolarity becomes 200 milliosmol, and now the urine which is going to be produced is going to be small in volume but having very high osmolarity of around 200 milliosmoles. Next, you can see the role of urea in forming concentrated urine. What happens is that your urea is freely filtered from the glomerulus into the Bowman's capsule. As it passes down the length of the nephron, most of it is reabsorbed, but once it enters the thick ascending limb and then the distal convoluted tubule and the collecting tubules and the collecting ducts, these portions are impermeable to urea, and urea is not reabsorbed and it cannot move from the nephron into the interstitium. And as you know that the late distal convoluted tubule and the collecting tubules and collecting ducts are highly permeable to water in the presence of ADH. When a lot of water is reabsorbed from these portions, the urea which is left behind becomes very concentrated, and as this concentrated urea moves down towards the last part of the nephron, this end portion of the nephron is highly permeable to urea. urea again, and this highly concentrated urea is going to be reabsorbed into the renal medullary interstitium, thus increasing the osmolarity of the renal medulla and helping the countercurrent multiplier in increasing the renal medullary interstitium hyperosmolarity, and some of it is going to be reabsorbed back into the loop of Henle. Again it is going to move along the length of the nephron. Again it is going to be reabsorbed from the last portion of the nephron. Again it is going to increase the osmolarity of the interstitium, and some of it is going to be reabsorbed in the nephron in the descending loop of Henle, and again it is going to move along the length of the nephron and again be secreted. So in this way recycling of urea helps in increasing the osmolarity of the renal medulla and also helps in formation of concentrated urine. So this is how your countercurrent multiplier and your urea recycling with the help of ADH, in the presence of ADH, help in the formation of concentrated urine. So finally, this hyperosmolarity, which was produced by the urea recycling and by the countercurrent multiplier and ADH, has to be maintained, otherwise it can be lost to the blood flow which is flowing in the peritubular capillaries. As you already know that when the nephron dips down into the medulla, there is also present a network of capillaries around this nephron which are known as the peritubular capillaries, and these capillaries are responsible for reabsorption of substances from the nephron. And what happens is that when the peritubular capillaries move from a dip down into the medulla from the cortex, they are going to gain solutes because the osmolarity of the medulla is high as compared to the osmolarity of the peritubular capillaries. Let me repeat, as you already know that the kidney can be divided into two parts, the cortex and the medulla, and the nephrons which are involved with the secretion of concentrated urine are known as JG nephrons, and they are mostly present in the medulla, and these are the nephrons which make the renal medullary interstitium hyperosmolar. So this is the reason that the medulla of the kidney is hyperosmolar. When the peritubular capillaries dip down into the medulla, the osmolarity of the medulla is very high, while the osmolarity in the peritubular capillaries is very low, and the solutes tend to move from the medulla into the peritubular capillaries, and as the peritubular capillaries move back from the medulla towards the cortex, they lose this solutes back into the interstitium, thus maintaining the osmolarity of the interstitium. Now let me explain again. Suppose this is a kidney and this portion is the cortex and this portion is the medulla, and you already know that we created hyperosmolarity in the medulla with the help of the JG nephrons, and we increased the osmolarity in the renal medulla to around 1200 milliosmoles to 1400 milliosmoles. And you also know that there is present a capillary network around the nephrons which is known as peritubular capillaries. The blood flowing in the peritubular capillaries has an osmolarity of around 300 milliosmoles because this is the normal osmolarity of plasma. So as the peritubular capillaries dip down from the cortex into the medulla, you can see that the medullary hyperosmolarity increases from 300 milliosmoles to around 1200 milliosmoles. So as the peritubular capillaries move downwards, the solute tends to move from the renal medulla into the peritubular capillaries, because the solutes are always going to move from higher concentration towards the lower concentration. So as the peritubular capillaries move down towards the medulla, solutes enter into the peritubular capillaries from the renal interstitium. So by the time the peritubular capillaries reach the deepest part of the medulla, they have gained so much solutes from the renal interstitium that the blood in the peritubular capillaries become as high in osmolarity as 1200 milliosmoles. So now there is a fear of this large amount of solutes to be washed away from the renal medulla, and there is going to be loss of renal medullary hyperosmolarity because the blood is going to wash away this large quantity of solute. But the nature has provided a very special mechanism over here which is known as countercurrent exchanger, and this is thanks to the U shape of the peritubular capillaries. Now what happens is, as the peritubular capillaries take a turn and start moving from the medulla towards the cortex, the osmolarity of the interstitium starts to reduce, because we know that the highest osmolarity is near the medulla, and the osmolarity of interstitium near the cortex is around 300 milliosmol. So as the peritubular capillaries take a turn from the medulla towards the cortex, they start moving from an area of higher concentration of solutes to an area of low concentration of solutes, and now the peritubular capillaries start to lose solutes from the blood back into the interstitium. So the solutes which they gained while moving towards the medulla are now lost as the blood flows from the medulla back towards the cortex. So there is no net loss of solutes, and no solutes are washed away from the renal medulla, and they are returned back to the medulla as the peritubular capillaries loops backward towards the renal cortex. And by the time peritubular capillaries reach back the cortex, they have an osmolarity of 350 milliosmoles. So they only succeeded in taking away 50 milliosmoles of solute as they pass from the medulla towards the cortex again. So the U shape of the peritubular capillaries helps in maintaining the osmolarity of the renal interstitium. Had there been a straight peritubular capillary, it would have washed away all of the solutes from the renal interstitium, and this would have left the renal interstitium very hypoosmolar, and the countercurrent multiplier would have to work again to create a hyperosmolar interstitium. But luckily the shape of the peritubular capillaries is a U, and this helps in the maintenance of the osmolarity of the renal interstitium, because the solutes which enter the peritubular capillaries on its way down are again sent back towards the interstitium on its way up. In this way there is very little loss of solutes in the peritubular capillaries. This is known as countercurrent exchange, and this helps in maintaining the hyperosmolarity of the renal medulla. So I hope you understood what is meant by the countercurrent exchange. Please do not confuse it with the countercurrent multiplier, because countercurrent multiplier was the whole mechanism which was involved with the formation of hyperosmolar renal medullary interstitium with the help of the thick ascending limb which had the sodium potassium 2 chloride co-transporter. So it is totally different from the countercurrent exchange. The countercurrent exchange takes place in the peritubular capillaries, and it is only involved with the maintenance of the osmolarity of the renal medulla, which was formed by the countercurrent multiplier. So remember that countercurrent multiplier forms the hyperosmolar renal medullary interstitium, while countercurrent exchange maintains it and does not allow solute which was accumulated with the help of countercurrent multiplier to be washed away through the peritubular capillaries. So Let us now move on to the next topic, which is the osmoreceptor ADH feedback system. So as I already mentioned that your ADH is formed by a special nucleus which is present in the hypothalamus, and this nucleus is known as supraoptic nucleus. Your supraoptic nucleus is involved in the formation of ADH. This ADH is then stored in the posterior pituitary, from where it is secreted into the blood. So let us now discuss how the ADH is secreted and what are the factors which control the secretion of ADH. So suppose here we have the hypothalamus and this is the pituitary, which is attached to the hypothalamus through its stalk. So in the hypothalamus there is present a special nucleus which is known as the supraoptic nucleus, which is involved in the formation of ADH. In the proximity of the supraoptic nucleus are present special receptor cells which are known as osmoreceptors, and they are involved in determining the osmolarity of the extracellular fluid. They are in close contact with blood vessels and extracellular fluid. So whenever extracellular fluid osmolarity increases, especially in the case of dehydration, there is increased solute concentration and decreased water content. This is going to result in increased osmolarity of the extracellular fluid. So these osmoreceptors, when they come in contact with this hyperosmolar extracellular fluid, they shrink and they send special signals to the supraoptic nucleus and prompt the supraoptic nucleus to increase the production of ADH. So there is going to be greater production of ADH from the supraoptic nucleus, and this ADH is transported to the posterior pituitary, from where it is excreted into the blood. Once ADH enters the blood, it acts on the distal parts of the nephron, especially the distal convoluted tubule and the collecting tubules and the collecting ducts, and it causes reabsorption of water from these later parts of the nephron. So let's repeat this. What I told you right now is that when there is increased plasma osmolarity, the osmoreceptors which are present in the hypothalamus, they shrink in size and they send signals to the supraoptic nucleus, which is also present in the hypothalamus, and this supraoptic nucleus is going to produce ADH in large quantities and transport it to the posterior pituitary, and this ADH is now going to be released from the posterior pituitary and enter the blood, and it is going to reach the nephrons and act on the distal parts of the nephron, especially the late distal convoluted tubule, the cortical collecting tubules and the medullary collecting ducts, and it is going to cause reabsorption of water from these parts of the nephron, and this reabsorption of water is going to reduce the extracellular fluid osmolarity back towards the normal. So this is how the osmoreceptor ADH feedback loop works, and this is how the osmolarity is maintained within the body. The hypothalamus, and they are quite near to the supraoptic nucleus, and these osmoreceptors have very rich blood supply. Whenever the blood becomes hyperosmolar because of dehydration, the osmoreceptors are going to shrink because they are going to lose water into the blood. As you know, water moves from a region of low concentration towards the high concentration. So water from the osmoreceptors is going to move towards the extracellular fluid, and the osmoreceptors will shrink, and this shrinking of osmoreceptors is going to send signals to the supraoptic nucleus, and supraoptic nucleus is going to form ADH and send it to the posterior pituitary, and from there it is going to be released into the blood, and this ADH is going to act on the late distal convoluted tubule, on the medullary and cortical collecting ducts and collecting tubules, and it is going to cause reabsorption of water from these parts of the nephron. And how ADH works on these proximal on these distal parts of the nephron is that when ADH, which is also known as AVP, arginine vasopressin, it acts on the receptors which are present on the basolateral side of the epithelial cells of the nephron. When ADH attaches to its receptors, there is stimulation of G coupled protein, and this G coupled protein converts ATP into cyclic AMP, which activates many protein kinases within the cell, and phosphorylation of multiple proteins takes place. As a result, there is production of aquaporins, which are basically channels which allow water to pass through them. These aquaporins, which are known as aquaporin 2, are incorporated on the luminal side of the renal epithelial cells, and they act as channels to allow water to enter from the lumen of the nephron into the renal epithelial cells. There is also incorporation of different types of aquaporins known as aquaporin 3 and 4, which are incorporated on the basolateral side of the renal epithelial cells. These act as channels for the movement of water from within the cell to the peritubular capillaries passing through the basolateral membrane. So this is how ADH helps in the reabsorption of water from the late parts of the nephron. So whenever there is water deficit, the extracellular fluid osmolarity increases, which is detected by the osmoreceptors. They send signals to the supraoptic nucleus, and the secretion of ADH is increased. Whenever there is increased ADH secretion in the blood, the water permeability from the distal convoluted tubules increases. Water is reabsorbed in large quantities, and very little water is excreted in the urine. This water, which is reabsorbed, causes the osmolarity of the extracellular fluid to decrease. So the osmoreceptors stop sending signals to the supraoptic nucleus, and no further ADH is formed. This is a negative feedback loop of osmoreceptor and ADH secretion. Next, let us discuss the importance of thirst mechanism in controlling your extracellular fluid osmolarity. So whenever your extracellular fluid osmolarity increases, not only is there secretion of ADH, but there is also activation of thirst mechanism, and as a result of this activation of thirst mechanism, you start feeling thirsty and you look for water and you drink water to quench your thirst, thus reducing the extracellular osmolarity of your extracellular fluid back towards normal. So let me explain. Whenever your extracellular fluid osmolarity increases, such as in dehydration, your thirst mechanism is activated. The centers for your thirst are present in your central nervous system, especially in the wall of the third ventricle and in the preoptic nucleus of your hypothalamus. So these centers are activated and they cause thirst in the person, and the person drinks water and the osmolarity is reduced back towards normal. So let us discuss some stimuli for thirst. Whenever the extracellular fluid osmolarity increases, or the blood volume decreases, or the blood pressure decreases, the thirst mechanism is activated. It is also activated when there is increased angiotensin II production, or when you experience dry mouth, when your mouth is dry. So all of these stimuli are going to cause thirst mechanism to be activated. When the plasma osmolarity is reduced, or blood volume is increased, or blood pressure is increased, or there is decreased angiotensin II, or when your stomach is distended, such as after a meal, the thirst mechanism is suppressed and you no longer feel the thirst. So the stimuli which decrease thirst are decreased plasma osmolarity, increased blood volume, increased blood pressure, decreased angiotensin II, and distension of the stomach.